Hi everyone, and welcome back to Biology 206. I'm Dr. Jennifer Baltzer, and today we are going to be talking about food webs and energy flow. There's a lot of carryover from previous lectures, so we're going to be returning to some of the same topics that we covered in our community lectures, but diving into a little bit more detail on things like food webs and trophic cascades and some of these really important interactions that involve not just the species in the community, but also the environment, which moves it from a community level question to an ecosystem level question. Okay, so with that, let's get started. Okay, so just to remind you, when we think about species within a community, we can group them by trophic level. And so the trophic level is effectively the feeding category of that species. And so in this diagram, we have our autotrophs. These are our primary producers. They are at the first level of the food web, okay? Or the first trophic level. The trophic level is based on the number of feeding steps by which a species is separated from autotrophs. So we have autotrophs here in the first position, and then we can move up through second, third, and fourth position in these trophic levels, with the second level being primary consumers, so herbivores, the third level being secondary consumers or primary carnivores, and then the fourth level being tertiary consumers or secondary carnivores, all right? So we have these different feeding levels that represent different trophic positions in an ecosystem, in a food web, and energy moves from this lowest level up to the highest level. So herbivores consume autotrophs. Those herbivores are consumed by primary carnivores. Those primary carnivores are consumed by secondary carnivores. And we're going to be talking about those energy flows through this system. Another important component of this is the fact that you see this red arrow that goes down. A huge amount of living biomass, of biomass from living organisms is not consumed. And in fact, those organisms die, and that organic matter is returned as detritus, all right? And this is where these guys come into play, the detritivores. They are feeding on a range of organic material in the form of detritus from various levels of the food web. And so we place those detritivores in the second position because detritus is considered as part of that primary position in the food web, okay? In the primary trophic position. So detritus is down here with autotrophs in the first position, or the primary position. And so detritivores are considered at the same level as herbivores, even though they are breaking down organic material from any of these levels, right? You can think about detritivores consuming dead animal and dead plant material, okay? So these are trophic levels. This is a little bit of review, but a little bit more detail than we've seen in previous lectures. And a really important consideration this is why I emphasize this aspect of detritus, it's a really important consideration of energy flows in ecosystems is the massive amount of detritus that is produced. Most organisms in ecosystems are not consumed but become detritus. You can think about, even if you think about something as simple as the litter that falls onto the floor of any forest each year, that's all living material that did not get consumed. It was not consumed by herbivores, and it falls to the forest floor and is broken down by various detritivores, all right? And that's just one example. Detritivores basically deal with any of the organic matter, the dead organic matter from plant, microbe, animals as well as feces from any animals, okay? And basically what happens is those detritivores fragment this organic material and help to move it toward remineralization. So you've got nutrients that are bound in organic compounds, and releasing those nutrients to become available for uptake by autotrophs again requires mineralization. You think about the nitrogen cycle, okay? And so the first step of that is just kind of the breaking up of that organic material so that it's more accessible by microbes, by fungi, etc., all of which are also parts of the decomposition process, parts of the dealing with detritus. So you have things like, here we have a sea cucumber. Things like, you can think of earthworms, you can think of millipedes and centipedes. These are all organisms that basically break down dead material, and then that dead material is can also is broken down further by microbes and fungi to be made available for uptake again by plants. And in some cases, fungi do not require that, fungi and bacteria do not require that breaking up of the organic material. In this case, we have these fruiting bodies of a saprotrophic fungus, so a fungus that decomposes wood, and so you can see all of these lovely fruiting bodies on the surface of a decaying log. Okay, so huge amount of energy is not consumed in across trophic levels and is returned as detritus and requires breaking down in order to become available again for uptake by autotrophs. And so there's this massive amount of energy that moves through living organisms and then and is returned to living organisms via detritivores. So they play a really critical role in these ecosystems. And again, this process is known as decomposition. Another component of detritus that's really important to think about is where the detritus is coming from. Okay, so when we think about energy flow in ecosystems, if a lot of the energy is coming through detritus, if you have outside sources of detritus, this can potentially lead to a greater availability of energy in the system. And so a great example of this is stream ecosystems, where you can see all of these overhanging trees, all of this litter and roots even extending into this stream bed. Those are all terrestrial sources of energy, of carbon, of nutrients, some of which falls into this stream bed and is available for breakdown by detritivores in the stream. And so we can think about sort of two different inputs of detritus or energy into ecosystems. One is allochthonous inputs, and these are external energy inputs. And so this example of the stream is a great example of that, where you have terrestrial inputs to an aquatic ecosystem. Okay, so you're moving from the forest ecosystem to the stream ecosystem, and there's terrestrial inputs from that forest ecosystem contributing energy to the stream ecosystem. Autochthonous inputs are energy inputs produced within the system. So if we think about this forest ecosystem, the litter that falls from the canopy onto the forest floor and is returned to that forest soil for uptake again by the trees is an autochthonous input in that system. So typically, terrestrial systems have a lot more autochthonous inputs, and aquatic systems have a lot more allochthonous inputs, particularly streams. Lakes are, it really depends on the size. I think it really depends on the size of the aquatic ecosystem and how much, in this case we see that these trees are completely overshading this stream bed. You can imagine, though, a large lake where very little of the lake area is impacted by the surrounding vegetation. And so that's a really important factor in how much of the energy inputs will be allochthonous versus autochthonous in aquatic systems. And it's important to know too that there are definitely some situations where we have the reverse happening, where energy from aquatic ecosystems moves into terrestrial ecosystems. And a great example of this, it's much less common, but a great example of this is in salmon migration. on the West Coast, for example, where juveniles go out to the ocean. They spend most of their lives there. They gain most of their energy in these marine environments, and then they migrate back upstream into terrestrial freshwater streams. And so that in and of itself, because we know that those salmon migrate up there, lay their eggs, and then the adult salmon die, that's an allochthonous input, a marine energy coming from the marine system into the terrestrial freshwater system. So it's an allochthonous input into that stream. But we also have the situation where terrestrial mammals consume those salmon that have marine, energy. Predominantly, the energy in those salmon is predominantly a marine sourced energy. So terrestrial mammals consume those salmon, and then that energy is brought into the terrestrial environment, because eventually those mammals will die in the terrestrial environment and be broken down. So you can see how, allochthonous, energy can be transferred across ecosystems in different ways. But when it's just important to keep these in mind, we sometimes have energy moving from outside the ecosystem to inside the ecosystem, or being transferred out of the ecosystem to a different ecosystem. And depending on whether you're aquatic or terrestrial, the importance of these allochthonous and autochthonous inputs differs. Okay, the next piece I want to cover is this idea of energy flow between trophic levels. And so this is going way back to high school probably for you folks, but the second law of thermodynamics is that during any transfer of energy, some energy is dispersed as unusable energy because of the tendency toward increased disorder or entropy. And so we can think about this in the context of trophic interactions. So a huge amount of energy is produced, is locked away in the bodies of primary producers. So this autotrophic biomass production, there's a huge amount of energy locked away in that, in both terrestrial ecosystems and aquatic ecosystems. The vast majority of energy is in the autotrophs. And you have herbivores feeding on those autotrophs, and a variety of processes that we'll get into in terms of efficiencies associated with both consuming, assimilating, and then producing biomass, assimilating energy from the lower trophic level, and then assimilating that into producing biomass from that. There's a lot of energetic loss associated with that. And so we move from one trophic level to another. There's a tremendous amount of reduction in the energy, both because of those losses associated with the second law of thermodynamics, as well as, in many cases, or at least in terrestrial environments, the reduction in biomass associated with the higher trophic level. Okay, so we have a loss of energy between trophic levels because of the costs associated with consuming, assimilating, and producing, consuming and assimilating energy, and then producing biomass from that energy. You can think, for example, of metabolic energy costs as one of those ways that a herbivore or any of these heterotrophs lose energy from this more energetically rich level of autotrophs. So these trophic pyramids that we see here, these are trophic pyramids. They show us how energy moves through the system. And so this can be, determined for any given ecosystem. And in terrestrial ecosystems, we tend to have, we can also look at the relationship between energy, which in both aquatic and terrestrial ecosystems, you have the most energy in the primary production level, in the first trophic level, and then that decreases as you move up through the trophic levels. For terrestrial ecosystems, biomass pretty well mirrors that energy, like the trophic pyramid for energy. And so we have the most biomass in primary producers, and then this decreases as you move up through trophic levels. This can be a bit different in aquatic ecosystems. In really resource-rich and nutrient-rich aquatic ecosystems, for example, where you have upwelling, you'll tend to have something similar as we see with the terrestrial ecosystems and have, energy and biomass being comparable because there's a lot of nutrients available to support a large autotrophic biomass, okay? But in nutrient-poor ecosystems, we may see what we see here, which is an inverted biomass, an inverted trophic pyramid for biomass, where there's a lot more biomass in each sequential level up from the autotrophs. And part of what is going on there is that in nutrient poor aquatic ecosystems, the turnover of phytoplankton. So we have a major difference between terrestrial and aquatic ecosystems in that the main primary producer in terrestrial ecosystems is, trees and larger vascular plants. This is where a lot of this biomass comes from. In aquatic ecosystems, it's very, it's unicellular phytoplankton. It's very small. It tends to be very small organisms that are much more simple compared to the autotrophs that are in terrestrial environments. And in nutrient poor parts of the ocean, the turnover of this phytoplankton is really high. And so you tend to have a fairly low biomass of primary producers. And the other component of this is that the efficiency with which higher trophic levels can take up resources from phytoplankton is much greater than it is in terrestrial environments. So if you think about terrestrial environments, we have, these fairly advanced plant bodies where there's, a lot of undigestible parts of the plant, where there's investment in chemical defenses and physical defenses against herbivores. It's much harder to consume resources in these terrestrial environments, to consume plants in these terrestrial environments than it is in aquatic environments where you have, fairly undefended phytoplankton as the autotrophs that are much energetically accessible compared to plants. And so herbivores in terrestrial ecosystems have to spend a lot more energy in digesting and consuming and digesting the autotrophs in these environments compared to aquatic ecosystems where the kind of nutritional fingerprint of the carbon to nitrogen ratio of tissues in phytoplankton is much more comparable to that of heterotrophs, and so much higher nitrogen compared to carbon, making it much more digestible, much higher quality, which can lead to a situation where you have a higher biomass of the higher trophic levels compared to lower trophic levels. Okay, so that's energy flow between trophic levels. And as a consequence of some of these differences in the way, kind of in the accessibility of autotrophs in different ecosystems, we can see changes in the relationship between net primary productivity, or the energy that's fixed by autotrophs each year, compared to the amount that's consumed by heterotrophs. And so in general, we see, so here we have NPP, net primary productivity, that's how much carbon is fixed per unit area per year by autotrophs. And on the y-axis, we have consumption by heterotrophs, and this is grams of carbon, again, on a meter square basis per year. So how much carbon is taken up per unit area per year. And in general, we see this positive relationship. If there's more biomass being produced in a landscape, there's more biomass being consumed in a landscape, and that makes sense. But when we look between ecosystems, so here we have a terrestrial ecosystem, here we have an aquatic ecosystem, there's an upward shift. The intercept of those lines differs, even though the slope is really quite similar. And part of the difference here, so we have, per unit biomass, higher consumption by heterotrophs in aquatic ecosystems compared to terrestrial ecosystems. If you just look at this value of 100, at an NPP of 100, we have, much lower consumption by heterotrophs in terrestrial ecosystems than aquatic ecosystems. And remember, this is on a log scale, so this difference is really quite large. And again, this has to do with the accessibility of the autotrophs. In terrestrial environments, these autotrophs are just harder to consume, harder to digest, harder. It takes more energy to convert plant biomass to heterotroph biomass. Okay, this is in contrast to aquatic ecosystems where the main autotrophs, which are phytoplankton, are much easier to consume and to convert into energy in the heterotroph, and so we have higher consumption in aquatic ecosystems than terrestrial ecosystems. So in terms of numbers, it's about 13% versus 35% of primary production is consumed in terrestrial versus aquatic ecosystems. So almost triple the primary production is consumed in aquatic ecosystems compared to terrestrial ecosystems. All right, and so this relates to this idea of trophic efficiency. So when we think about trophic efficiency, we think about, okay, you've got your net primary production, that's the amount of carbon that's fixed per unit area per year. And as we talked about before, a whole lot of this is not consumed and becomes detritus to be broken down by detritophores, right? Some portion of this is consumed. So the consumption efficiency is the proportion of available biomass that's ingested by consumers. So as we saw on the previous slide, the consumption efficiency in aquatic ecosystems is higher than the consumption efficiency in terrestrial ecosystems. So you have consumption efficiency. Some of that is lost to feces and urine. You know, we excrete various nutrients and energy in our feces and in our urine. And then this is then handled by those detritophores, as we already talked about. So the amount of the available energy that's consumed that actually makes it to this assimilation phase is part of that consumption efficiency as well. Okay, so then we move to this assimilation efficiency, and so we've lost. So okay, our consumption efficiency is how much of this total proportion of NPP is consumed by the next trophic level, by the primary consumers. Part of this is lost to feces, part of it is assimilated, and so the assimilation efficiency is the proportion of the ingested biomass that is that consumers assimilate by digestion. So the feces and the urine are not assimilated by digestion. Some proportion of that energy then is assimilated by digestion. And that assimilation efficiency is a function of the quality of the food that you're consuming. So we can think about this terrestrial aquatic comparison where the phytoplankton have a much lower carbon to nitrogen ratio. So they're much more nitrogen rich compared to carbon, whereas terrestrial environments, plants tend to have much more investment in lignification and different defenses, makes it really hard to digest them. So that can lead to a difference between those two kind of main ecosystems. The assimilation efficiency can differ between different trophic levels. So the quality of food available to carnivores is much higher than the quality of food available to herbivores or detritivores. So carnivores are eating other animals. Nutritionally, those animals are much more easily assimilated compared to the plants that a herbivore might eat, or the decaying biomass that a detritivore may eat. And so the assimilation efficiency of those three different categories, it's 80% in carnivores versus 20% in herbivores and 50% in detritivores. Okay, and detritivores are higher than herbivores in part because they're also consuming some of those more readily accessible resources like animal bodies. Okay, so differences in assimilation efficiency. The other thing is that endotherms tend to, when we think about assimilation efficiency, we think about the digestion efficiency. Okay, so endotherms tend to digest their food more completely compared to ectotherms, and in fact, in some species, there's specialized features in the digestive system that help them to break down food, and this can include mutualistic relationships with different gut biota that allow them to more effectively break down the food that's coming into their system. Okay. All right, so that's assimilation efficiency. And then the third piece is production efficiency. So what proportion of that assimilated biomass is used to produce new consumer biomass? And this comes down to metabolic costs. And so when we think about metabolic costs, how much is lost to respiration, right? When we think about metabolic costs, it's some obvious places where groups diverge is, again, endotherms versus ectotherms. Endotherms have to spend an awful lot more resources keeping their body warm, so they have to generate metabolic heat. And this is very costly and reduces the amount of biomass that can be produced. Similarly, large organisms versus small organisms. If you have, otherwise comparable organisms, a large organism is going to be more efficient, and its production efficiency is going to be higher because its metabolic rate is lower compared to the smaller organism, right? A larger surface area to volume ratio that you have in a smaller organism means that it has to generate more metabolic heat, its metabolism has to be higher to function compared to that large organism that has a higher volume to area ratio. Okay, and so here's just a table that looks at those production efficiencies. And so you can see birds and small mammals have fairly low production efficiency. Only 1.5% of the assimilated energy is converted to consumer biomass. Compared to about 3% in large mammals. And then if you go from these endotherms to ectotherms, it's really quite incredible. Fish is accepted. We've got somewhere between 20 and 60% of the energy that's assimilated being actually converted to biomass of the consumer when we're talking about ectotherms. So big differences there. And trophic efficiencies are important when we're thinking about population dynamics. Of course, the food resources available to species are not necessarily static through time. And as those food resources change, the various trophic efficiencies can change and impact the population in beneficial ways or in harmful ways. And in this case, we're seeing an example where these sea lions from the Gulf of Alaska, we have this lovely picture of these guys here. They had a very large population from, the 60s to the 80s when they were being monitored. That was up, north of 250,000 individuals. And then we see this precipitous decline. And by the year 2000, the population was down to 50,000 individuals. Researchers are really concerned, well, what is happening to this particular population? Other populations of sea lions were doing fine. They looked at the time, they thought perhaps the prey species were declining. They looked at the amount of time that sea lions were spending searching for, out hunting for prey. And the sea lions in the declining population were not spending any more time than the sea lions in the static population. So it was not an issue of, the fact that prey was hard to find. So that was not the issue. But then they took a look at the contents of scat and sea lion stomachs. When a sea lion died, they would open up the stomach and take a look at what was inside. So they had scat information and stomach content information from different time periods. And so here's back in the mid-70s when the population was really at its peak. The mid-80s, it had started to decline, and the mid-90s when it's pretty close to its low, all right? And what we can see is that the types of prey species that the sea lions were able to access was changing, or that they were accessing was changing. So in the early 70s, we see there was very little consumption. We see primarily that sea lions are consuming these small schooling fish, so herring, capelin, eulachon, species like this. These tend to be fairly energetically rich species. You think of many of these species, they're fairly oily fish. They're fairly energetically rich. As we move through time, we see that the types of prey that they're targeting is changing. We see a decreased reliance on these small schooling fish, an increased reliance on cod and pollock and hake, cephalopods, so squid, and an increasing reliance on various flatfish. Okay, so they're still accessing lots of food resources, but the food resources are changing, and this changes again when we head into the mid-90s. And so when we think about, you could do, if you measure the quality of these different prey species, you can evaluate how much, how much energy is available from these different prey species, and consider the efficiencies of these different prey species for sea lions. So the researchers speculated that that this shift in the feeding in the prey species was impacting the trophic efficiencies that they were, the trophic efficiencies in terms of how much energy was actually being able to be put into production of biomass. And this is leading to, less healthy adults and lower reproductive outputs compared to populations that were able to maintain this this historic diet. There's some other factors, and your text gets into that, but this is just an example where we can think about how changes in the population can impact, the changes in the populations of prey can impact the populations of the predator via these trophic efficiencies. Okay, another topic we have touched on is the idea of trophic cascades. We're just going to dive a little more deeply into this, but if you remember, when we talked about trophic cascades, we talked about this idea of, or we talked about an example, a really well-documented example of declining sea otter populations. So on the one hand, there was over-harvesting of sea otter populations for their pelts. On the other hand, typical prey species of killer whales was declining, and the killer whales were becoming more reliant on sea otters, driving their population numbers down. So we can talk about different ways that trophic cascades can happen. One is through top-down control, and this is the rates of consumption at higher trophic levels may dictate abundances and species composition at levels below them. And so that example of the sea otters is a great one. So if we imagine, top-down control, we have our secondary carnivores, in this case it would be that killer whale, that shifted its focus to sea otters, killer whale or humans, shifted its focus to sea otters because its own prey species were declining. Sea otter population, the sea otters are a primary carnivore, the sea otter population started to decline, and so they were no longer feeding as heavily on the sea urchins, which are the herbivores. That herbivore population exploded, the sea urchin population exploded, having a direct negative impact on the primary producers, the kelp, that form those kelp forests. And so this is a great example of a trophic cascade, a top-down control in a trophic cascade, okay? Bottom-up control is where the abundances of these higher level, so series of changes in abundance and species composition, resources that limit NPP determine the energy flow through the ecosystem. So NPP is determined down here at the primary production stage. And so really in bottom-up control, you have some limiting resources. That could be light, that could be nutrients, that could be water availability, that dictates the primary production. And that primary production, the level of primary production then dictates the abundances of all of these subsequent trophic levels. All right, so we've got bottom-up control, we've got top-down control, okay? And so these trophic cascades then are a series of changes in abundance and species composition. And we just had an example of that with the sea otter example. Another example from your text of top-down control comes from New Zealand. And so very early in the colonization of New Zealand by Europeans, Europeans decided to introduce brown trout, which was a native species, to their homes, and sort of the idea was that by introducing this species, it would make them feel a little bit more at home. And so these brown trout began being introduced in the 1860s in New Zealand, and by 1920, about 60 million fish have been released into streams and lakes. And so the introduction of a non-native species into these native environments. So it creates an interesting situation for trying to understand trophic dynamics when you bring in a new predatory species. So the introduction of these brown trout led to declines in various native species. And in this graph, the native species is Galaxius. And so what we're seeing here is some work that was done by a couple of scientists, Flecker and Townsend, where they looked at the impact of these brown trout on, first of all, invertebrate densities. So the brown trout and the Galaxias both prey on invertebrates in these stream and lake ecosystems. And so what we see is when there are no fish present, we have the highest densities. And so this is invertebrate density in numbers per meter squared, so population size per area. Remember that oftentimes invertebrates are measured with little quadrats, and so we can estimate their density per area. So we have no fish having the highest density of invertebrates. With Galaxias, the density of invertebrates was lower than with no fish, but it was lowest when you had this invasive brown trout in the system. Okay, so brown trout was putting much more pressure on the invertebrate population than was the native Galaxias. Okay, so not surprisingly, you add this predator to the system, and the primary consumers decline. So then what we see is a change in the primary producers. So you can measure chlorophyll A as a measure of chlorophyll A, of course, is a key pigment in chloroplast, the photosynthetic, the organ of photosynthesis in plants and algae. And by measuring chlorophyll A in water, you can measure, it basically gives you a measure of the abundance of algae, okay, in the system. And we can see that when there's no fish, the chlorophyll A concentration is fairly low compared to Galaxias, where it's a bit higher. It's about double that of no fish. And then brown trout, it's the highest. And so we see this top-down control of primary productivity as you introduce this additional predator into the system. Invertebrate density reduces. This impacts the amount of herbivory on algae in the system, changing, leading to an increase in algal productivity in this particular ecosystem. Okay, so another example of top-down control. Now, you may wonder, okay, well, in most of these examples, we're only seeing, up to sort of 4 levels, 4 trophic levels. And so, what determines the number of trophic levels? How many trophic levels can we have? Okay, and so we have some food webs where we have a very, very simple trophic structure, maybe with only, the primary producers and the primary consumers, so a 2-level trophic structure. Maybe a predator comes in and consumes that primary consumer. Okay, so now we have a 3-level trophic system, but if that predator is lost, then we revert to a 2-level trophic system, or if the predator reestablishes itself, we move back up to a 3. You can also lose a consumer, you can lose or gain a consumer from an intermediate trophic level. And whether or not you lose then this, the level above it is determined by whether this individual can consume the level below it, okay? So in the event you lose a consumer and it's the only consumer that supports this higher trophic level, then you could lose both of them from that system, right? And you can also modify the way trophic structure works, especially when you're thinking about something like an omnivore, where the food preference of an omnivore can change, and so this can impact the pressure on one or another part of the trophic system. Okay, so an omnivore could increase its pressure on some level of consumer. An omnivore could increase its pressure on primary producers, and so the dynamics within those trophic networks can change through time as well. Okay, so there's a number of factors that can influence the number of trophic levels. So the amount of energy entering the system. If you have more energy in an ecosystem, there's greater potential to support viable populations of higher trophic levels, okay? If you have a system that has very low energy inputs, it's likely that you're not going to be able to support a really complex trophic structure. The frequency of disturbance can also be an important factor. We talked about the intensity and the frequency of disturbance in our last lecture. And so how frequently a disturbance comes in can be really important in determining whether you end up with these higher trophic levels or not. All right, so for example, if you think about a wildfire burning through a system, initially everything's gone except a bunch of organic matter, and you have to have the reestablishment of vegetation. The reestablishment of vegetation eventually will bring in those primary consumers, the herbivores. And then once that herbivore population builds up sufficiently as the autotrophs continue to grow, eventually you'll be able to bring in some consumers, some carnivores into the system. If you disrupt that system too soon, it will not get to a place that it can actually support those higher trophic levels, okay? So the frequency of disturbance can be really important for determining how complex your trophic structure will be. And finally, the ecosystem area is really important. Where you have small areas, first of all, you have, we know that the area available determines population size. So a smaller area will support only a smaller population because of carrying capacity. And a smaller population we know is more likely to go extinct. So a large area can support a large population that is going to be more stable. And so in a large area, we're more likely to be able to support viable populations across more trophic levels compared to in a small area. The other component of it is that in a large area, we have a greater diversity of habitats available, which then can also provide greater support for a higher number of species. So when we, again, thinking back to a lecture or two ago where we talked about species area relationships, the larger the area you have available, the more species, the more likely you are to have more complex trophic structures. Okay, so there's a number of factors that dictate how many trophic levels, and that's important to think about when you're contemplating, energy flow in the system and the complexity of these species interactions. All right, finally, moving back to this idea of food webs. So everything is connected to everything else. We've already seen food webs. Here's a very simplistic version of a food web from a desert ecosystem. You have, again, these autotrophs, and we saw a portion of this at the start of the lecture. We have these autotrophs producing food for grasshoppers and ground squirrels. Those grasshoppers and ground squirrels provide food for scorpions, and then for golden eagles and kit fox. But that's not the whole system. There's lots of other species, and so as we add more species, we see more complexity, more linkages, more trophic levels, in fact. And so we have, plants and ground squirrels and grasshoppers. So we have our primary producers, our primary consumers, but then we have these scorpions that consume grasshoppers, but also can be consumed by these roadrunners and shrikes. All right, shrikes and roadrunners can also directly feed on the primary consumers. And then finally, we have some added complexity here in the top predator level, because we now introduce this species, the great horned owl, which feeds on the roadrunners. And you can blow this up even further to be the whole kind of the whole system. And here we have our plants, our insects, our arachnids. There's a whole diversity of things that fall into each of these trophic levels. And there's a huge amount of complexity in terms of the connections between them that very rapidly becomes quite overwhelming. Some things to highlight are the fact that you can have, one trophic level feeding on a lot of other trophic levels. So I'm just trying to find an omnivore in here, but we have some omnivores in this system that feed on both plants and also feed on other animals. And then we have also these U-shaped connections where we actually have cannibalism happening within a particular trophic level, within a particular species within a trophic level. So as you add realism to your food web, it becomes very, very complicated very quickly, and it's really hard to interpret these energy flows. So these more simplistic diagrams really help you to sort of understand what the key energy flows are in an ecosystem. One way that we can start to think about, which are some of the key. So again, if we go to this, it's really hard to look at this and kind of come up with any idea of what are the really important links. There are so many linkages here that it becomes overwhelming. And so one of the things we want to try to do with these food webs is identify what are the critical links. From a conservation perspective, we want to know which of the species in here, if we lose them, are really going to have a dramatic impact on this food web as a whole, okay? And so this is where we get back again to this idea of interaction strength, okay? So a measure of the effect of the population of one species on the size of the population of another species. This is really important. Identifying interaction strength is really important because it allows us to focus our attention on important connections in a food web, okay? One way we've talked about this is through removal experiments. If you can experimentally remove a species from a system, you can evaluate its impact on that system or on a particular interaction by measuring the abundance of a population of its prey species or of one of its predators. If you remove that species, what are the implications for the populations of species that rely on it or upon it which it relies? These are really not very feasible in many places. It's really hard to just eliminate species from systems and evaluate the impact. So there are other ways that people do this. So feeding preference studies, where you evaluate which species are feeding preferentially on which other species, and couple that with information on population dynamics. If you know the population of one species and the population of another species, you can look at how related they are to one another. You can imagine back to those graphs of coupled predator-prey cycles we've looked at, like the lynx and the hare, where their population cycles are so tightly connected. If you remove one from the system, the other is going to have a really big problem. And so if we know something about the feeding preferences, we can then couple that with information on population dynamics of these connected species and evaluate how important one is for the other. By identifying these really critical links or keystone species, which we learned about in our last lecture, this can help us understand those complex webs with big conservation implications, okay? So we can then start to identify, okay, what are the critical linkages within these food webs that if they're removed, that whole food web or portions of it are going to fall apart. So a great example is has to do with this starfish, and there's a number of invertebrates that it feeds upon, and an observation was made that when starfish were removed, the diversity, the richness, and the diversity of the population of invertebrates upon which that starfish fed decreased. Okay? So we would see a decrease in species richness of the invertebrate community in response to a removal of starfish. And what these researchers found, the researcher was Robert Payne. He hypothesized that these starfish were playing a pretty important role in controlling the populations of certain invertebrates. So he hypothesized that at least one or more of the invertebrates was a better competitor than the others. When the predation pressure from the starfish was removed, those competitive dynamics were able to play out more clearly. And so indeed, what happened was when starfish were removed, these mussels were able to really overtake a lot of the space. So a lot of these are sessile invertebrates. So they affix themselves to hard substrates in the intertidal zone. And so the mussels were actually able to outcompete a lot of the other species for space in this environment. And by doing that, reduce the richness of the invertebrate community as a whole. When starfish were present, they kept those mussels in check. Interestingly, when the starfish were removed and the mussels were allowed to sort of overtake the system, and then the starfish were reintroduced, the system had actually reached a point of hysteresis. If you recall our last lecture when I was talking about hysteresis, even though that predator was returned to the system was returned to the way it was before the removal of the predator, the mussels had gotten too large to serve as prey species for the starfish. And as a consequence, the system did not recover following the reintroduction of the starfish. So it entered a new stable state, if you remember that conversation we had about stable states. Okay, the final piece of this is thinking about complexity of food webs and whether that enhances stability. For a long time, early ecologists thought that complexity in a food web should enhance stability, because the more connections you have, the more potential there is for redundancy. The weaker the connections are between one species and another. So if a species is lost from the system, it's not going to have as big of an impact. Some modeling work done suggested that there was kind of mixed support for this. If you in some early modeling studies, it pointed toward the fact that even though there were more connections, and maybe those connections were weaker because of these many more interactions in the food web, the loss of one species could kind of propagate through the food web in unexpected ways and lead to reduced stability. But one way that greater complexity, certainly some studies have found that where you have greater complexity in certain parts of the food web, it can lead to stability. So if we think about plant species diversity, if you have a more diverse plant species community, you have more stable biomass. So you have species that are able to respond differently to different environmental conditions. And if one declines, herbivores still have a number of others that they can rely on. And so you can think about those components of diversity as promoting stability in a system. Likewise, if you have diversity of herbivores, and the predator can feed on a number of different herbivores, then even if a herbivore population declines, another herbivore population can support the predators that rely on it. And so this is what is referred to as a portfolio effect. The variation in population of one species cancels out that of another, leading to stability. So where you have kind of more diverse systems, even if the interactions are not necessarily more complex, that greater diversity in some of these trophic levels can support stability of the food web as a whole. Now, the last thing I want to end on is a really important idea that many of you will have heard about. Some of you may have even read this book by by Rachel Carson. It's called Silent Spring. Rachel Carson was really the founder of the environmental movement, and she identified the fact that these ideas of bioaccumulation and biomagnification, which relate directly to energy exchange across trophic levels and food webs and all of these ideas we've just been talking about. So in her book Silent Spring, Rachel Carson outlined the impact of a chemical pesticide that was used in a very widespread way in the 60s and 70s, DDT, even into the 80s. This chemical was a DDT, and it was sprayed to control various insects, including forest pests, including mosquitoes at beaches, etc. And she documented the bioaccumulation of this toxin. So she demonstrated that organisms were able to take this toxin up from their environment. So you spray DDT, it ends up in the water. organisms take this up. So bioaccumulation is the gradual concentration or accumulation of toxins in an organism through its life. Okay, so all of us have toxins in our bodies that we've been accumulating through our lives by eating various things and by different chemicals that we've been exposed to. Biomagnification, and this is what Rachel Carson talked about in Silent Spring, is this idea of biomagnification, which is the concentration of those compounds, these toxic compounds, across trophic levels. So lower trophic levels ingest the compounds, and they're in their bodies at some concentration. And then a higher trophic level ingests a lot of that lower trophic level, and all of the toxins from all of those many individuals at the lower trophic level get incorporated into the next trophic level, and so on. And this leads to what we refer to as biomagnification. And so in the case of DDT, and so you can see these pictures, this is a crop sprayer spraying DDT over a forest to control insect pests. This is DDT being sprayed, and you can see this little boy right in the cloud of the DDT, which we now know is a terrible toxin, being sprayed to control mosquitoes at a beach, of various biting insects at a beach. Okay, so this was used in a really widespread way, no protection on the people doing the spraying, people in the path of the spraying, no thought as to what the potential implications were. And Rachel Carson really identified that this was what happens, where you have, DDT being sprayed into the environment, some portion of it ends up in the water system, okay? Producers take this up. They take up the water with the DDT, and that DDT is concentrated in the tissue of the producers through their lifespan. Those producers are consumed by zooplankton, so small primary consumers, small planktonic consumers. The DDT in the tissue of the producers is concentrated in those zooplankton, and so you can see the concentration of DDT being magnified as you move up these trophic levels, because the DDT is not metabolized or excreted by these organisms, and so it's concentrated in their tissues. And so it continues to be concentrated until it's at an incredibly high level in these fish-eating birds. And the way that the impact was identified was that the eggshells of these birds, basically the populations of bald eagles, were collapsing because they could no longer, their reproduction was not viable. DDT was leading to thinning of eggs and reproductive failure in this species. And once DDT was no longer sprayed, the populations of bald eagles rebounded. Okay, so this is a really fairly recent example of biomagnification in systems and the real consequences of this. So this was from, I remember this from when I was a kid, and a more recent concerning impact is the bioaccumulation of various persistent organic compounds in Arctic ecosystems, for example, and there's some information about that in your text, as well as the concentration of mercury. So mercury is deposited atmospherically, so various industrial processes release mercury. It's deposited atmospherically into ecosystems, and there are certain forms of mercury, specifically methyl mercury, that are toxic to life. And what we see in this graph is the bioaccumulation, the biomagnification of mercury in the tissues of various organisms in a pond system in Czechia, in the Czech Republic. Okay, and so you can see here we have mercury in micrograms per kilogram. We can see the concentration in the sediments and the aquatic plants and the herbivores. It stays pretty consistent in this category. We see it increases a bit here in the omnivores. So these are species that are feeding on one another, but also on the herbivores and the aquatic plants. And then we see it quite a lot higher, particularly in the cormorants in the system. So we see this massive increase, particularly in the cormorants, of mercury concentration in these top carnivore levels. And we're seeing this throughout Arctic ecosystems as well, where we see biomagnification of mercury concentrations leading to levels of mercury in fish tissues that are considered unedible by Health Canada standards, for example. So it has direct ramifications not just for ecosystem function, but for human health and well-being, where you have populations that are relying on these food resources. Their food resources are being threatened, and so the really big implications of the transfer of pollutants through trophic levels in these ecosystems, and we don't really understand. There's lots of pollutants that we don't have a good handle on, whether they biomagnify or what that looks like and what the implications are, despite the many, many chemicals we put into the environment. And with that, I'm going to finish the lecture. We will be moving on to the landscape unit after this, and so we'll be once again scaling up. But that's all I have to say about food webs. We'll be moving on now from food webs and trophic interactions to the larger scale of the landscape. Okay,